Good morning, ladies and gentlemen. My name is Rimawati Sukmana. First, I'd like to thank the organizing committee and Professor C for inviting me to speak in this meeting of the Vietnam Society of Sleep Medicine. I will share my screen now. Okay. My talk today is on sleep disturbances linked to genetic disorders. So genetic disorders or genetic factors are surmised to regulate sleep as evidenced by the heritability of sleep traits, specific genetic polymorphisms of these traits, and existence of familial sleep disorders. Studying the genetic factors underlying sleep disturbance will aid in understanding the underlying mechanisms of sleep. Identification of the first familial circadian phenotype, the familial advanced sleep phase syndrome, in the late 1990s, have begun genetic mapping and cloning of genes or mutations that have strong effects on circadian timing, thus starting the quest for understanding the genetics of sleep. Now we talk about the genetic factors of sleep. Genes regulating sleep remains ambiguous up till now. Why, we don't know yet, but sleep is not one phenotype. There are variabilities in REM and non-REM sleep. Sleep is a very complex behavior regulated by two processes, the circadian rhythm or process C, and the homeostatic drive or process S. Circadian rhythm sets the time of day that sleep preferably occurs, or sleep timing. Homeostatic drive or process S keeps track of prior sleep-wake history, controls homeostatic need for sleep, and sleep need will accumulate during wakefulness and decreases during sleep. So homeostatic drive will set the duration of sleep and the quality of sleep. This is the components of the mammalian circadian clock. The mammalian clock consists of 3 feedback loops of clock genes. The first one and the main one is clock BMAL, one mediated transition of, and it was mediated by the repressor pair and cryptochrome, period and cryptochrome genes. And there's another feedback loop, also the clock BMAL mediated transcription of the gene DBP and ROR, REF, ERB. And there are 3 distinct systems that regulate the circadian clock. Both processes, process S and process C, actually are genetically regulated. So the differences between process C and process S are not clear-cut. For instance, clock genes, the NPAS2 and clock, are sleep-wake driven, not circadian mainly. So clock genes also act as sensors of homeostasis in peripheral tissues. Also, sleep deprivation can cause long-term dampening of clock genes expressions. Variation of PAD3 is associated with sleep deprivation, and period 1 and period 2 and DBP genes are implicated in sleep homeostasis. And also, process S is caused by build-up and decay of adenosine, and the gene ADORA1 encodes adenosine receptor, and gene ADK encodes its metabolism enzyme. So both processes are genetically regulated. And also other genes also are associated with sleep. Voltage-gated potassium channels, like in Drosophila, we found that they found that sleep inhibiting effects of mutations in the Shaker potassium channel. Genes of the immune system also are associated with sleep. Interleukin-1 and TNF-alpha play a role in sleep physiology, and nuclear factor kappa B increases after sleep deprivation. And the gene relish in Drosophila has a role in controlling the NF-kappa B. Genes regulating can also regulate duration and timing of sleep. Like this gene, DEC2, the mutation in this gene carriers showed higher delta power during non-REM and less REM sleep compared with the non-carriers. Also, slow wave activity in non-REM sleep, theta and alpha activity during wakefulness and REM sleep were all increased in the mutation of period gene. And now we come to the sleep disturbances with genetic factors underlying narcolepsy and other hypersomnia disorders. Narcolepsy is a chronic neurologic sleep disorder that manifests as a difficulty in maintaining continuous wakefulness and sleep. It is caused by a marked reduction in neurons in the hypothalamus that produce orexin or hypocretin. The causes are complex genetical and environmental factors. In narcolepsy type 1, it's caused by deficiency of hypocretin and 1 and 2. These are the neuropeptides controlling wakefulness and appetite produced by the neurons in lateral hypothalamus. Probably it was triggered by an autoimmune pathology. Nearly all narcolepsy type 1 cases carry HLA DQB1 star 0602 gene allele, and also 30% to 50% of narcolepsy type 2 cases. There's also increased prevalence of narcolepsy type 1 was discovered after H1N1 infection in Chinese children and after anti-H1N1 vaccination in European countries. Single nucleotide peptide was also found between CPT1B and CHKB. CPT1B regulates beta oxidation. It's a pathway involved in regulating theta frequency during REM sleep. And CHKB is an enzyme that metabolizes choline, a precursor of the REM and wake regulating neurotransmitter acetylcholine. Also, idiopathic hypersomnia is presumed to be genetically caused by genetic mutations. The frequency of the QB10602, around 40% in idiopathic hypersomnia, is intermediate between that in the general population, 12 to 38%, and that in narcolepsy cataplexy, 70 to 100%. So it may be closely linked to narcolepsy. So the polymorphism is located between CPT1B and CHKB, may be associated with both narcolepsy and hypersomnia in Japanese cohorts. Kleine-Levin syndrome is a rare disorder characterized by episodes of hypersomnia, cognitive and behavioral changes. It's usually found in adolescent males of Jewish descent and other relatives. The probable major susceptibility gene and exposure to an unknown environmental trigger has been found. Now we are talking about circadian rhythm sleep-wake disorders. It's caused by alterations of the circadian timekeeping system, entrainment mechanisms, or a misalignment of the endogenous circadian rhythm and the external environment. Advanced sleep phase is characterized by a phase-advanced circadian sleep-wake rhythm relative to local soral time. Advanced sleep wake phase disorder is defined as a phase advance of sleep-wake cycle accompanied by a sleep-related complaint. Familial advanced sleep phase syndrome is a subtype of advanced sleep phase. It overlaps with ASWPD, and most cases of young onset ASP were familial. If FASPS, the patients tend to sleep and wake very early, and their melatonin and core body temperature rhythms are advanced by 4 to 6 hours, and their circadian rhythms are 1 hour shorter compared with conventional sleepers. These patients have defect in phosphorylation of period 2 gene, and so there's a mutation in period 2 and CK1 delta. Delayed sleep-wake phase disorder have later sleep and wake onset. They usually have sleep deprivation due to sleep onset delay by biological clock, and morning waking time is enforced by alarm clock and social or work responsibilities. Length polymorphism in period 3 and period 3 promoter is linked to this disorder. And also Patke et al. Found a mutation in the core circadian clock gene cryptochrome 1. Cause is sensation product to consecutively repress the circadian transcriptional activators. Now, there's also genetics of restless leg syndrome. Restless leg syndrome, this disease is a feel of urgency to move legs occurring with abnormal leg sensations that are relieved by movement. They have circadian presentation that's worse at nighttime. The imaging shows decreased striatal dopamine transporter and D2 dopamine receptor binding potential. Pathophysiology may be caused by iron dopamine connectivity, iron regulation in brain, opioid signaling, and brain and spinal cord circuitry. 60% of RLS patients report family members with this disorder, and 83% in monozygotic twins. It was found that the gene MEIS1 is dopamine signaling. It's expressed in dopaminergic neurons of substantia nigra, spinal cord, and red nucleus. And part of the regulatory network that specifies motor neuron full identity and pattern of target muscle connectivity. RLS is associated also with mutations in BTBD9 gene. It concerns striatal activity associated with periodic movement in sleep. Other genes are also associated with RLS, such as MAP2T5 and SKOR1. The SKOR1 is expressed selectively in subset of dorsal horn interneurons in the developing spinal cord. Insomnia also has genetic factors. Insomnia is a persistent difficulty of initiating or maintaining sleep with corresponding daytime dysfunction. Insomnia runs in families and is higher have higher concordance in monozygotic twins. Gene-wide association studies found 57 loci for self-reported insomnia symptoms. The examples that they have found is the MEIS1, CYCL1, TMEM132E, and SCFD2. They have also, insomnia has also shared genetic factors with RLS, aging, cardiometabolic, behavioral, psychiatric, and reproductive state traits. They've also found a possible causal link of insomnia with coronary artery disease, depressive symptoms, and just subjective well-being. Fatal familial insomnia is a severe sleep disorder with dysautonomia, motor sign, abnormal behavior as other symptoms. They have found a primary atrophy of selected thalamic nuclei and inferior olives, expansion to other regions with disease progression. It is linked with autosomal dominant inherited prion disease caused by mutation of the prime protein gene PRRP D178N. Loss of sleep, sympathetic hyperactivity, flattening of vegetative and hormonal circadian oscillations are the symptoms of this disease. Short sleeper, where they wake up at extremely early hour in the mornings, and sleep onset is comparable to conventional sleeper, but the sleep duration is 2 hours shorter than the conventional sleeper. In the short sleepers, they have found mutation in DEC2, which is a repressor of clock RB-mal activity, and it is also a transcriptional repressor of hypocretin 1 and 2. Sleep-related breathing disorder also have genetic origins, as RBG is abnormalities of respiration during sleep that may experience repetitive episodes of decreased or arrested respiratory airflow during sleep. Examples are OSA, central sleep apnea disorders, sleep-related hypoventilation disorders, sleep-related hypoxemia disorders. OSA is a repetitive upper airway obstruction caused by reduced patency of the upper airway during sleep. It is heritable. First-degree relatives are more likely to snore or have observed apneas after controlling for obesity, age, and gender. OSA have there is evidence of direct and indirect genetic distributions in OSA. Indirectly, the obesity, craniofacial structure, neurological control of upper airway muscles, sleep, and circadian rhythm have indirect genetic associations with OSA. 40% of variants in apnea-hypopnea index are due to familial factors. There's possible risk loci identified, which is associated with inflammation, hypoxia signaling, sleep pathways. They have found polymorphism in these genes. So OSA is caused by a combination of many genetic and environmental factors. Undine curse or central congenital hypoventilation syndrome is a life-threatening disorder. They have impaired ventilatory response to hypercarbia and hypoxemia. They have alveolar hypoventilation and autonomic dysregulation. This disease has mutations in the PHOX2B gene, the FOX2B gene. This is a master gene for formation and function of neuronal network for autonomous control of ventilation. Psychiatric disorders, they have found SNPs in core circadian clock genes associated with psychiatric disorders. There are several SNPs also in and that's associated with higher recurrence rate of bipolar disorder, major depression, and sleep disturbances. And schizophrenia, the gene period 1, period 2, period 3, and NPAS2 in white blood cells, mutations have been found, and it will cause disruption in diurnal rhythms. In schizophrenia, they have found loss of rhythmic expression of cryptochrome 2 and period 2 in fibroblasts. Also, altered clock genes may play a role in sleep problems, and the emergence of symptoms present in certain psychiatric disorders. Also, insomnia symptoms are associated with MACE1 in animal models. Depression symptoms that include early morning awakenings and fatigue is also associated with several genes. And also 80% of schizophrenic patients have sleep disturbance and caused by either exogenous or endogenous cause. The endogenous cause is related to pathophysiological processes on sleep continuity and architecture. And several genes have been associated with antipsychotic induced suspect syndrome, clozapine induced somnolence, and insomnia symptoms in schizophrenia. There's also sleep disturbances in neurodevelopmental disorders. They are probably multifactorial, genetic, neurobiological, and environmental factors may have the interplay here. These are the symptoms, the syndromes, and several symptoms of sleep disturbances that occur in these syndromes. So in conclusion, sleep is a very complex behavior influenced by genetic and environmental factors. However, the sleep genes have not yet been found. Most sleep disturbances are caused by interplay of genetical, environmental, and biological factors. So understanding the genetic factors underlying sleep disturbance may help us to discover the mechanisms of sleep. Thank you very much.